Hi, my name's Tim Levine. I am working at the University College London Institute of Ophthalmology, which is over in the city and about 2 miles away from the rest of UCL. I researched a fundamental issue about how cells work. So every single living element on the planet is made of cells, and so we are millions of cells in each direction, but you also unicellular organisms. And each cell has a very similar body plan within it. And these body plans are often described in the way that microscopy reveals it to us. So the sort of thing people working in cell biology, like me, looking at how individual cells work, is looking at all the different constituent parts of the cell. And these constituent parts are often best defined by microscopy, which shows all the internal gubbins within cells. And they're all surrounded by little membranes. So the field is called membrane trafficking. And if you look at what Google says about membrane trafficking, it shows all the interesting little parts within cells, and it gives them all nice shapes. Sometimes it draws them as special footballs or stacks of plates, or lots of little small vesicles. I hope you can see these small things. But one of the things that none of these diagrams show is how these things within the cell actually relate with each other physically. It always shows them at some distance between each other, as if there's lots of white space, gaps. Here's another one, huge gaps between the things that are of interest. But of course, this is not how the cell is. There's no gaps at all. Everything's crushed right up against each other. There's no space. And this is not described in textbooks, but what really happens is a lot of these things, which are called organelles, very much like organs in a body, inside the cell there's organelles, they all touch each other and they come into contact and they pass material physically directly from one place to another. And that's my interest, is how these organelles touch each other. And the material I'm particularly interested in is lipids, things like the fats that go in to make olive oil or cholesterol. How are these moved around inside cells? It turns out it's almost all by direct contact between, for example, where they're made and where they're going to end up, and then they jump across very small spaces, and that's what I work on. My interest with RITs came about because I realized that it's very hard to work out what sort of proteins might sit at these places where two organelles touch. To know what they would be like, we'd need to know what sort of structures they have. But we know very little about a lot of proteins. Most proteins we don't know very much about. We don't know their structure. And there was a piece of software which I realized had been invented many years ago, but it took until about 2011 before I realized how brilliant it was, that could be used to predict the structure of proteins and maybe find new members of the field I'm interested in, proteins that sit at these contact sites. And I started using the software bit by bit, but I realized I needed to do it across the whole of an organism, every single protein in an organism, to actually get a good grasp as to what's going on. And that project required much more computer power than I could possibly do on my own. And also, the second half of the project was to make a website so that anyone could look at these results, and they would be in a presentable form and also downloadable as well, so that people who came across a hit could then access the raw data themselves. So that is what different parts. So I had help from two different people in UCL computer department. One who helped me run 20,000 searches, producing gigabytes of data, and the other providing a platform on which they can be viewed online. So if I could just take you through an example. This is not a perfect example because, and you'll see why. It starts off with a human problem, which is SARS, CoV-2, coronavirus, COVID-19. Okay, so lots of research in COVID-19 has gone to provide huge numbers of potential targets inside cells. So this is a map of all the proteins in humans that allow a human cell to multiply the virus. You can see here, this is zoom in. All these proteins have really annoying names. Some are more annoying than others, like this, TMM41B. This means nothing. This just means it's a protein with a transmembrane domain. We don't know anything about it. So using the website Help with RITS, I have come up with some great insights on that protein. But I'll just walk you through this one. This is another protein of completely unknown function. So I'll show you, if you go into Google and ask this protein, what is known about this protein is very little, unknown function. This is the sort of protein it's related to in yeast. And I've only found this by using the HH yeast database made for me by UCL Computing. And its domains are, it's a very long protein. It's got over 1,000 residues, and it's identified as being a member of having domains in it that are basically itself. All that's known about it is that it is itself, and nothing is documented about what itself really is. But using the HH yeast server, I can link it to this crystal structure, 3BUT, and it's very clear what that is. That's a lipid transfer protein. So this is exactly the sort of protein I was looking for all along, that is involved in lipid sensing and lipid traffic inside cells. So using this work on yeast, I can then transfer it back to humans and show that this protein, which I showed you here, involved in the production of coronavirus in humans, also I can look at its function and make a very good guess what residues are important in this protein, so we can make testable hypotheses about all sorts of cellular functions, including those related to SARS coronavirus 2. Okay, well, that's a really good question. So in the past, we managed to do 20,000 searches, and we just covered the proteome or the genome of a small model eukaryote, budding yeast. And we made a website. But what we'd really like to do is cover the whole of the rest of eukaryotic and maybe even bacterial evolution by scaling this up. So unfortunately, bioinformatics websites do not involve this piece of software. They stick at much less sensitive software and leave quite a proportion of proteins undocumented. And applying the software that I can, with the help of UCL Computing, we can reduce this unknown by a considerable margin, I guess about 30 or 40%. So at the moment, I'd say 15% of the proteins are unknown. And with the application of this software now to a much wider group of organisms, including humans, so that would more directly, more obviously identify these two hits as what I think they are, we could reduce this from 15% unknown to about 10% unknown. And so I'd like to get further funding as a result of the success of this first project to roll it out to maybe 20 times the size of the first project. So use that as a sort of pilot.